The previous slide has some information about starch digestion that's important, but I don't think I need to comment further than what's on the slide. This is a really important slide here on the villus. The villus are those finger-like projections that go into the lumen of the small intestine, and they are loaded with enzymes to fully break food down. Sometimes they're called the brush border enzymes. The brush border is referring to the microvilli that are on those epithelial cells. The epithelial cells are what make up the villus. Inside the villus, you have a capillary bed, and so when you're absorbing um, the nutrients go into the epithelial cell, so they have to go in, and then they have to go out the other side to get into the capillary bed. The lacteal is an important structure. It's actually part of the lymphatic system, and fats go in there. Fats are too large to go into the capillaries, so fats are absorbed actually into our lymphatic system. Believe it or not, they go into that lacteal, which is usually shown as a green-like structure in the middle of the villus. The intestinal glands that you see there, they're also making enzymes. So we have enzymes from the glands, enzymes from the brush border. Mucus is also made. I remember having a professor jokingly say that if it wasn't for the mucus that lines our entire digestive tract, we'd be screaming every time we ate. And so, like the stomach's lined with mucus, small intestine, everything's lined with mucus to help protect it from the enzyme attack. The following slides talk about absorption. And I think I mistakenly used the word reabsorption on the very first slide. That reabsorption term is actually used in the kidney, and I sometimes mix up the terms. Here we're doing absorption, and IB wants you to know a couple different examples for each type. So think back to that first unit where we talked about how things go across the membrane. So we got simple diffusion, and the examples I'm giving come right off the study guide because there's lots of examples and you can't remember them all. So simple diffusion, fatty acids. Facilitated diffusion can also be fatty acids, evidently. That happens both ways, or fructose you can use for that one. Active transport would be mineral ions such as sodium, calcium, and iron. So what we're talking about going into the epithelial cell, right? And then a lot of these mechanisms are also used to get the stuff out of the epithelial cell into the capillaries or the lacteal. Um, the very last slide shows a nice graphic of lipid digestion, which is quite complicated. And so the picture has a really nice diagram. And on the slide that says objective 12, I wrote once fatty acids and monoglycerides are absorbed, the triglycerides are made in the epithelial cells. They're coated with a single layer of phospholipids, cholesterol, and protein, and are called lipoproteins. They are released by exocytosis. So they're released by exocytosis out the other side, and then they go into the lacteal.